our lecture. I think we have already 24 people joined for your lecture, so it's very high interest to your lecture, which is great. And first thing, I would like to introduce our speaker for today. This is Dr. Stepan Stehlik. Dr. Stepan is a senior researcher at the Institute of Physics of the Czech Academy of Sciences, where he leads the nanodiamond and interfaces group. His research is dedicated to nanodiamonds, with a focus on understanding how their origin, size, structure, and surface chemistry govern their electronic and interfacial behavior, with relevance for environmental, energy, and biological applications. Stepan has authored more than 50 peer-reviewed publications and has coordinated or participated in several national and international research projects on hydrogenated, quantum-sized, and doped nanodiamonds. In recognition of his scientific contributions, he receives the Otto Wichterle Award of the Czech Academy of Sciences. So, Stepan, please tell us your story of your research of nanodiamonds.
Okay. So let's try it. So I hope that the screen is visible. So thank you very much for a very kind introduction, Olga. And I have to thank you and the Adamas Nano company that you gave me this opportunity to present our latest research on the hydrogenated nanodiamonds, and specifically on their surface charging and overall interfacial properties. So at the beginning, I would like to introduce a bit our environment. So I'm from the Institute of Physics of the Czech Academy of Sciences, and we are located not at the main site, but at a bit hidden campus in Prague, which is a division of the solid state physics, a very nice historical campus and overall nice place. And we belong to the Department of Semiconductors, where we have like two bigger clusters. One is nitride semiconductor and theory teams, and the second one is our diamond team, which we formed together also with my colleagues Alex Kromka, Gabovanko, and Stepan Potocký. And we have quite long tradition actually since 2003, where a first CVD deposition system has been installed. And currently we have a nice CVD lab equipped with 3 systems. Fourth one is on the way to us, and we also have a nice new labs for the processing and characterization of the nanodiamonds and of course other samples. So historically, or maybe a few words about our research strategy. So we of course balance between the fundamentals and also applications. So recently we did a lot of research in the size-related effects in nanodiamonds. We wanted to clarify what is the true phonon confinement, for example, in the nanodiamonds, which is a phenomenon in the Raman spectra. So that's what we published a few papers about that. And but these size effects are also important in the colloidal properties and electronic properties, like you will see today. And recently we moved more to the surface chemistry and electronics, and it's definitely related to the hydrogenated surface, which is, as you will see, very interesting and worth to study. And regarding the application directions, we are involved in some bio-oriented studies and in the quantum sensing. Here we have a collaboration with Petr Ziegler and Michal Gulka, on the NV centers, but we can also produce our own silicon vacancy doped nanodiamonds from the CVD films. We also published some nice works in the energy and environment field, like some photocatalysis on the titanium nanodiamond composites for decomposition of organophosphates gases, very poisonous ones. Also showed that the hydrogenated nanodiamonds can serve as an electron transport layer in the organic photovoltaic cells, and also very recently collaborated on the work showing that these composites, titanium nanodiamonds, can also serve as a nice separators in the supercaps. And because we have many or several CVD deposition systems, we are also very interested in the seeding and overall in the CVD growth. So we also published very interesting papers here, like the using of a size-reduced 2 nanometer detonation nanodiamonds for very dense seeding and growth of ultra-thin films. Then also we achieved some very nice porous morphology of boron-doped films. And recently we also showed that the quality of the seeding is also influencing the properties of the resulting diamond films, which is also very important result in my opinion. Okay, but today I will speak mostly about the charging of hydrogenated nanodiamonds. I will introduce the hydrogenated nanodiamonds as a kind of counterintuitive interfacial system and show when does this diamond-like behavior actually emerge. Of course, I will show that not all the nanodiamonds are the same, and especially focus on the nitrogen as so far unknown factor with a huge effect on the interfacial properties. And in the end, I will stop by at some implications for real interfaces and applications. So let's start with the reference case, which is for us, or must be, I would say, the hydrogenated bulk diamond, which is a well-known surface, and it has been studied for some time, and it is very specific. I think it's widely known, but I think it's important to repeat that it has a negative electron affinity, which causes this enhanced emission of electrons from the surface. And because of the transfer doping effect, where the electrons can actually escape the material and get localized on some surface adsorbates, the holes which are left behind are quite mobile. And so there is this p-type conductivity, which is sensitive to the environment. You can make a nice transistors, field effect transistors for some power electronics and very fast transistors, but you can also use it as a sensor because, for example, as a biosensor, because of the nice biocompatibility of the diamond in general. So this conductive channel can somehow sense the some processes relevant to cells, for example. What is also important to mention that the surface is hydrophobic, and the CH bonds which are on the surface are actually like from the classical point of view not ionizable, but we see that they can, or the surface can exchange the electrons. And regarding the colloidal properties, it has been measured that this hydrogenated bulk diamond has a negative zeta potential. So this is very important for today's talk, and it was explained by this scheme that actually here is this potential drop from the positive to negative, and the negative potential measured as a zeta potential is coming actually from this negatively charged adsorbates, which could be, for example, OH minus ions. But when we turned to the hydrogenated nanodiamonds, we suddenly see exactly the opposite thing. And suddenly we we see the strongly positive zeta potential in aqueous environments. So it has been demonstrated, I believe, for the first time in 2007 for hydrogenated diamond powder that time. But since then it was reported many times, and I think it's widely known that the hydrogenated nanodiamonds have quite robust colloidal stability, but still without these classical ionizable groups. Over the time, there were some attempts to interpret or explain this behavior, and they were, I believe I can identify like two approaches which have been or which appeared in the literature. One approach was, or one approach assumes the protonation of electron-rich sp2 surface. So this study actually supposes that this positive zeta potential is because of this protonation of the sp2 surface, but there is nothing about the role of the hydrogen termination actually. And the other models, they, on the other hand, they suppose this hydrogen terminated diamond-like transfer doping, but however, it's difficult to, let's say, compare it with this bulk surface and with its negative zeta potential. So you can see that there is a quite, I would say, mess, or it's not clear which parameters leads to which properties. So our aim was to shed a light a bit on this phenomenon. I also want to point out that this positive zeta potential and overall the hydrogenated nanodiamonds are also quite perspective in some applications, like their positive surface charge can immobilize some valuable biological relevant proteins. It can also remove the fibroblast growth factors, for example, because there is a strong affinity of the fibroblast growth factors towards the hydrogenated nanodiamonds. So it can be used in some therapy of some diseases where this FGF expression is too much. And also it has been shown by Ollie Williams and colleagues that it's also very useful for the filtration. So it can capture some negatively charged contaminants, including viruses. But also their electronic properties and photo properties are interesting, like it has been shown quite recently, very useful photocatalyst for the PFAS decomposition, just 2 studies at the moment. This is our contribution I've already, and this one I've already also introduced. Okay, so let's now define our playground, our nanodiamond landscape. And because as you will see, this comparison of different types of nanodiamonds is very important. It's actually our approach, how to distinguish the effect of the individual parameters. So I think everybody knows the HPHT nanodiamond and the detonation nanodiamond. So these 2 classes are actually quite different. And in case of the HPHT nanodiamond, they are created by milling of the synthetically grown HPHT monocrystals. So all these shard-like particles are single crystalline. And if they have any surface sp2 carbon, it is located only on the surface. So they have random shape, they are polydispersed and broadly available. And I have to say also that they contain just very low content of the nitrogen in general, let's say. On the other hand, we have the detonation nanodiamonds, which are very defective particles. I call them internally polycrystalline because nearly every particle is like a twin, or you can see some defects. And the sp2 carbon is not limited only on the surface, but it can be found also along these grain boundaries and also in some regions inside the particles. Plus to this, they contain 2% of the nitrogen, which you will see is very important and so far not appreciated that much. But we can also identify some extreme cases which are very helpful in our studies and understanding. So on the HPHT side, the bottom-up HPHT nanodiamond can be grown. I will introduce this a bit just in a moment. But also on the DND side, we can also think of some new synthesis routes and precursors, which potentially, and you will see it's true, should give us some control over the composition and also over the size. But for today's talk, the composition is the important parameter. So let's have a look on this first part. When does the diamond-like behavior actually emerge? And for this, to answer this question, I would like to introduce a bit more this bottom-up HPHT nanodiamonds, which are created by the HPHT method directly from the molecules. And the very nice precursors are halogenated adamantanes, like it has been shown some time ago by Evgeny Yakimov, who actually provided us these samples. And what is very nice about this technique is that we can nicely control the size of the nanodiamonds just by the parameters and especially by the synthesis temperature. So with the increasing temperature, we can increase the size of the nanodiamonds, and this is a perfect, and these particles are really quite monodisperse, and as such, they are perfect for these size-dependent studies. What is also very special and very nice is that from the synthesis, you can see on FTIR spectra that they are perfectly hydrogen terminated. So here we don't need to do any ex-post hydrogenation like we do, but these particles from the synthesis have a perfect hydrogen termination. So when we investigated their properties like Raman and so on, we also investigated this size dependence of the colloidal properties. So we tried to prepare some colloids by sonication in water, and we actually realized that when we have the size below 2 nanometers, we were not able to get any nice colloids like we are used to with the hydrogenated nanodiamonds. But when we exceeded the size above 2 nanometers, suddenly we got quite reasonable colloids. So this blue is the Z average size, and this black one is the zeta potential. And also the zeta potential became positive only above 2 nanometers. So the zeta potential magnitude was not that high. It was about 30 millivolts, and then became quite stable. But so then we tried also dispersibility in something non-polar organic like chloroform, and we saw the opposite trend, and we saw a nice solubility even of these sub 2 nanometer particles, or sample, I would say, because it's not a particle actually, and very bad solubility or dispersibility of these bigger particles than 2 nanometers. Here are the TM images showing that the morphology is indeed different. So below 2 nanometers, we were not able to distinguish the particles. So the sample looked like kind of sheets or something. But over 2 nanometers, we started to see the individual particles. So it looks like that for the formation of the positive zeta potential, we need a nanocrystal of diamond, at least of 2 nanometer in size, and it's quite probably relevant to the formation of the electronic structure that at least 2 nanometer is required to be able to have these properties relevant to the hydrogenated diamond surface. Okay, so I told already something about what actually generates the positive charge, but let's have a look by which attributes it is also modulated. So indeed, then we tried to analyze the electron structure of our samples. And here it is a nice comparison of hydrogenated DNDs and hydrogenated high pressure, high temperature nanodiamonds. This time, these are these milled commercial particles. And in both cases, we saw after hydrogenation, we saw quite nice reduction of this electron affinity. But on DND, it remained positive. But on HPHT nanodiamonds, we really achieved a negative electron affinity, just like on the single or bulk hydrogenated diamond. So this is the proof that on these hydrogenated nanodiamonds, we have the driving force caused by the hydrogenation for this, let's say, effects that are known for the bulk hydrogenated crystals as well. But it was not only that. We also observed some dependence on the particle structure. So you can see here that the magnitude of the zeta potential actually correlates with the particle structure and especially the non-diamond carbon content. So these bottom-up HPHT nanodiamonds, as I have said, were about 30 millivolts. And from the Raman spectra, you can see that they are very pure. So it's just diamond and some residual peaks from the CH bonds, actually. The top-down HPHT nanodiamond, it's a still single crystalline, but it has already some sp2 carbon. And DND is a well-known Raman spectrum with a strong non-diamond carbon content, and DND has the highest zeta potential. So it looks like that these two factors are in the game. The hydrogenation is needed, of course, and there is also this dependence on the non-diamond carbon content. What else we compared was the electronic activity, especially the conductivity. So we showed that both HPHT nanodiamonds, this bottom-up and top-down, were quite conductive compared to the DNDs, which were non-conductive, or at least 5 orders of magnitude lower conductivity. So it's also correlating with their negative electron affinity and the positive electron affinity, but also maybe there is some influence of the structure and nitrogen whatsoever. This we don't know yet. But what is also interesting, and I want to show that in FTIR spectra, we can observe this Raman-like feature as a negative peak at the Raman frequency. So we saw this peak only in our HPHT conducting nanodiamonds. And in the spectrum of DND, we saw a peak at this frequency, which we normally see, and I will also get to that later. But what is important now is that it looks like that this kind of transmission window is somehow connected with the conductivity. And I was quite happy that this paper from Ivan Vlasov's group appeared some time ago, and it explains this feature, because we observed that, but we were not able to explain it. But this paper appeared and explained that it's coming from the phenotype interference between the phonons. That's why it's the phonon frequency, the discrete state of phonons, and the continuum of the charge carriers. So actually, this feature to appear, it signalizes that we have some charge carriers inside and or on the surface or close to the surface, and it's quite nicely correlating with the conductivity. Okay, and the other parameter which we found is different is the hydration. So you can see again on the FTIR spectra of the freshly deposited samples that we see quite common OH signal on the DNDs. We saw something also on the HPHT top-down in this case, but we saw nothing on the bottom-up HPHT nanodiamond. So the affinity to water here was the lowest. And it was also confirmed when we annealed the sample, we still saw the OH and the water on the DNDs, but not on these HPHT types. I have to say that this persistent hydration of the hydrogenated DNDs is quite well known. It has been also maybe first time showed by Korobov. Also some our work appeared that we also noticed this difference hydration already some time ago. And also nice work of Tristan Petit, also showing this different hydration and some strange things happening in the hydration shell. But here our results indicate that this is actually quite specific to the detonation nanodiamonds only. Okay, so to quickly sum up this initial part, so we compared the HPHT nanodiamonds and the DNDs, and we saw quite different behavior. So here we see the negative electron affinity, the conductivity behavior. We saw the weaker zeta potential and weaker hydration, and we saw in IR this negative funnel-like feature. But in DNDs, we saw the positive electron affinity, insulating character, highest zeta potential, very strong hydration, and we saw a peak at this Raman frequency. So that was the state like 2 years ago, and then we realized that we would like to disentangle which parameter is actually responsible for this very specific behavior of detonation nanodiamond. But then we realized that we have still 2 parameters which differ these 2 particles, and it's the nitrogen content and the structural defects. So then we fortunately started collaboration with the company OZM, which is the Czech company which is now involved in the detonation synthesis of the nanodiamonds, and they provided us a very special sample. It is the DND sample, but with a low nitrogen content. So it has about 10 times lower nitrogen content than the classical DNDs, and which were also provided by them. So these are made from the TNT RTX mixture, like typically. And we also involved the single crystalline HPHT nanodiamonds as a monocrystalline reference. So these are the newest results I'm going to show now, not yet published, but hopefully close to the acceptance, the paper. Okay, so when we did the TEM, we noticed that the internal structure is quite similar in these 2 DND samples, which was something we wanted. So the only parameter, hopefully, which is different here is the nitrogen content. Okay, so let's compare. Also, let's briefly go through some fundamental physicochemical characterization. So the Raman spectra, you can see that this low nitrogen DND is indeed a bit different than the DND material. It has slightly less shifted and slightly narrower diamond peak. And also there is some difference in the G band, where we see more a higher contribution of this graphitic-like band and or component, and lower contribution of this 1590 band, which is associated probably with some isolated sp2 defect, and this one is very prominent in DNDs. And here we have our single crystalline HPHT nanodiamond. So the nitrogen difference is quite visible from the XPS spectra, where we see small but still a very, very persistent peak and very common of the nitrogen in DNDs, but here we don't see any peak basically. And the complementing XRD analysis just showing a very similar defective structure compared to the single crystalline HPHT particles. Okay, so then we wanted to investigate their properties as a function of the hydrogenation temperature. So we made a very broad hydrogenation range. So we hydrogenated the particles in between 500 up to 1300, just in order to have some incomplete hydrogenation, some optimal hydrogenation, and we also wanted to have some graphitization actually. And that's why we went so high. And we followed we measured the residual mass, which is very simple but quite interesting parameter, the diamond to G-band ratio from the Raman, also oxygen and the nitrogen. So this is the residual mass. So you can see here that initially we see a decay of the residual mass, which is associated with the hydrogenation. It's a replacement of the original heavier oxygen on the surface by lighter hydrogen and formation of the CH bonds. But when we go higher with the temperature, we exceed some stability limit, and we see the graphitization which is connected with the mass loss, because the hydrogen is still a bit reactive atmosphere, so the particles are actually gasified. And this is quite correlative, the Raman intensity diamond to G-band ratio. So here we see in the beginning a nice prominence of the diamond, but then some sp2 carbon is formed as a result of this thermal hydrogenation. It's also quite well known, but when we exceed to 1100, we get a severe graphitization. So you can see that the standard DNDs are a bit less resistant to the temperature than this low nitrogen DND, which behave more like this HPHT nanodiamonds. Okay, and this is also the oxygen content as a function of the hydrogenation temperature. So with the increasing hydrogenation temperature, we see in DNDs quite steep decrease of the oxygen, but then the oxygen kind of stabilize on 2%. Just this is not oxygen inside, but on the particles you will see. And in case of HPHT nanodiamond and low nitrogen DNDs, we got stabilization at about 1%. So our results are quite consistent with, for example, this one of Takeshi Kondo's from this paper, which I like very much. And this is just to demonstrate that nitrogen difference between the DNDs about 2% and the low nitrogen DNDs at about 0.2%, but just this here, the sensitivity of XPS is already very low. So these data are only like quantitative, sorry. Okay, so let's have a look now on the FTIR spectra of fully hydrogenated samples. And we were very interesting how the spectrum of the low nitrogen DND will look like, whether it would be like similar more to DND or to HPHT nanodiamonds. And it turned out that it's very similar to the HPHT nanodiamond. So you can see quite similar CH stretching pattern. We didn't see any prominent hydration, and not many or not strong signal in the fingerprint region. But in contrast, and this is quite common in DNDs, we see also the CH stretches, but a lot of additional features, a lot of other peaks, which are, as you will see, relevant to the water, to the hydration, and also carbon dioxide species. So this is this part and also this part. So let's have a look on it in more details, and let's get back to this Raman-like peak, which is really a very interesting peak, which we always see in the hydrogenated detonation nanodiamond, and we always, or up to now, assign this peak to the embedded nitrogen, but in a kind of a physical way, that the embedded nitrogen, because of the higher electronegativity than carbon, can induce some dipole moment in the diamond lattice, and that's why suddenly it's visible for the IR radiation. We also had a nice analogy or nice paper to cite in this regard. So it is this paper of Lawson where they showed this appearance of this Raman-like peak in the nitrogen-doped diamond, and they assigned the appearance of this to single substitutional nitrogen after positive charging. So we always thought that we have this peak because of the nitrogen and because of some similar effect. But then we made the desorption study by drifts, and it was measured in argon flow at different temperatures. So we wanted to desorb all the surface water, and we noticed that we also desorb or we also lose this Raman-like peak. So you can see that there is a nice dependence on the temperature. That in the beginning we see the increase of the intensity and then decrease of the intensity and nearly complete disappearance, and also some shifts of the frequency, which are, in my opinion, not very correlating with this origin, but it indicates some chemical origin, and the reason is the adsorbed CO2 from the air, which we confirmed by this experiment when we compared the ambient equilibrated sample, that's the green spectrum, with this Raman-like peak, and also with this little sharp peak, which is also coming from the CO2. And then when we equilibrated, or when we rehydrated the sample but without CO2, we saw just the OH bonds, but not restoration of this peak, and also not this peak. So then we further confirmed the presence of the CO2 by the mass spectroscopy, by the desorption study. So the green, and also of the water. So on DNDs, the green signal is the water signal. So you can see that the desorption occurs at about 150 or a little bit less. But then we have some second contribution, and simply some water is present up to 300 degrees C. So the hydrogenated DNDs are very hydrophilic, and they keep the water up to very high temperatures, actually. And also there is connected the CO2 signal, so you can see that the CO2 is dissolved in this hydration layer and goes out with this water up to 300 degrees C. In contrast, on the HPHT nanodiamond, we saw just some weakly physisorbed water, which was gone up to 120, and no CO2 signal. So it's obvious that this strong hydration and CO2 binding is associated with the nitrogen in the DNDs. And here I need to take credit to the group or the work of Aleksensky, who actually showed this CO2 capture some time ago already. Well, not that much. And they also made some exchange of the ions. So they proved that, for example, this Raman-like feature is coming from the adsorption of the bicarbonates or hydrated bicarbonates. And this interpretation was based on this nice paper of Garant, where they measured FTIR spectra of hydrated bicarbonate molecules. And when we found this paper, we suddenly got explanation for all these strange additional peaks we saw in the IR spectra of DNDs. So, for example, this little sharp peak here, which we also ascribed wrongly to some free OH on some hydrophobic interface, it's quite probably actually the OH in the bicarbonate, OH vibration in the bicarbonate molecule. Then the always present residual carbonyl, is also from the sorbed bicarbonate, and the Raman 1332 or similar frequency is also quite probably the stretching CO vibration, and its frequency is actually changing as a function of this number of hydrating molecules. So that was exactly that behavior during the annealing. So we also observed this frequency shift, quite probably as a function of a number of hydrating molecules. So it looks like that we have a chemical origin of all these additional peaks, especially this Raman-like feature. But let's finish this sub-IR story, which I could not insist to present. And let's have a look on the HPHT nanodiamond again. If you noticed, in contrast to the previous slides, we didn't see any transmission window at the Raman frequency here. So this is the reason why which we still don't understand much. We already have some clues, but what we saw was that when we deposited these conducting nanodiamonds, we saw this transmission window appearing on the gold, but we didn't see it on the aluminum substrate. So this was very interesting, and so this is in bigger magnification. So we believe that it's because of the different work function of these two metals, and it looks like that the nanodiamonds provide the electrons to the gold, and that creates this whole carriers which can do the interference. But on gold, on the aluminum, pardon. On aluminum, this doesn't occur because of the higher work function and gold and different equilibration. Okay, so that's all for the IR, and let's summarize it like this: that on DND, this Raman-like feature has a chemical origin which is associated with the nitrogen and absorption or attraction of the hydrated bicarbonates. But on HPHT nanodiamond, we have a physical origin of this feature, and it's associated with the charge and charge transfer. And now let's move to maybe the most important part, which is the charging of the hydrogenated nanodiamond. So here, this is important graph showing the evolution of the work function for these three nanodiamonds as a function of the hydrogenation temperature. So it's actually as expected, and also as indicated by the previous study. But what is nice that we made this kind of dependence as a function of the hydrogenation temperature. So we recorded this insufficiently hydrogenated surfaces. Here we saw some optimum, and here we saw again increase of the work function, which is quite probably because of the graphitization at these highest temperatures. What is very important here is that it looks like DND is the most reactive, and the work function goes down already at about 500 degrees C, even though we still have about like 6% of the oxygen. But both these low nitrogen nanodiamonds, they need a higher temperature to achieve the full hydrogenation, and the full hydrogenation is achieved at about 700 or 800 degrees C. Okay, and let's complement it with the zeta potential, and that's the result. So when we start with the DNDs, just 500 is enough to have this very high zeta potential, and then the zeta potential is kind of stable and decreases only at this highest temperature when the graphitization is really strong. And what is interesting is that these low nitrogen nanodiamonds, they develop the zeta potential like slower, let's say. So they need the higher hydrogenation temperature, and in both cases we see some maximum of the zeta potential, which is like at 1000 for low nitrogen DND and 1100 for the HPHT nanodiamond. And this is exactly just before the severe graphitization occurs. So this graph tells that not only the hydrogen termination is important to achieve the positive zeta potential, but its magnitude is also somehow associated with the surface structure and quite probably with the formation of this disordered sp2-like surface carbon content, which is highest just before the excessive graphitization. So then we constructed this unified picture, let's say. So it has 3 panels, and in the panel A, we simply wanted to show that the formation of the positive zeta potential is primarily because of the hydrogenation, as it indeed lowers the energetic barriers and it enables the charge redistribution towards the surface states. Okay, and so this is the general mechanism which we think is quite plausible. Here, we believe that there is indeed the protonation of this electron-rich surface or interface. This is still not perfectly clear. So it's actually like merge of those 2 models I've showed in the beginning. So this driving force is the hydrogenation, but also the sp2 surface or some disordered surface and the surface states, because of this, are also very important in the acquisition of this positive zeta potential. Why we think that there is the protonation? Well, it's we don't have any direct proof for this, but I think it's quite logical that when we measure the zeta potential, we always measure somewhere at this slipping plane, right? So the particles, we don't measure the neat particles, but some particles with some hydration shell. Otherwise, we would not have a stable colloid. And we saw that also the HPHT nanodiamond have some water on them. So we suppose that there must be some hydration shell, and the kind of ions which can cause the positive zeta potential are these hydronium ions. So this for us is the most plausible explanation. But it's also modulated by the surface chemistry. I'm sorry, by the surface structure, like I try to show here, that we observed the maximum just before the graphitization. And this is exactly the sp2 character of the surface, which enables this charge localization at the surface or at the interface, which is in contrast to ideal hydrogen-terminated diamond, where the charge is actually transferred to some external acceptors. And I've already showed that these properties are modulated by the nitrogen, that the nitrogen actually enhances strongly the hydration and also the interfacial properties, especially towards the CO2. And this is just to show the recent paper of Arzen, showing this presence of these surface states, which may probably, quite probably, play the role in this localization or of the charge at the interface. Okay, so let's finalize with some implications. So I think this work is quite relevant to the recent work of Taro Yoshikawa, who introduced this very nice material, I would say, material, and it's a boron doped layer where there is a the porous DND nanolayer of about 50 nanometers, where they suppose that because the DNDs here are the classical ones, nitrogen rich, so it actually enable to behave like a photoactive nanolayer. So it's really working for the CO2 photoreduction, and the function of the DND is believed to work as light absorption and also electron excitation via the nitrogen inside the particles. And from our perspective, we think that also the interfacial properties can play a role. So because we show that the nitrogen is kind of a chemical amplifier, it also provides the higher zeta potential and stabilizes the hydration and concentrates the CO2. So it's quite possibly also play the role in this approach. But however, I have to say that this surface basicity of these nitrogenated nanodiamonds, it's not yet clear whether it is some chemical effect, like we know from the sp2 carbon. When they are doped with the nitrogen can be behave like a local base, or we can also think of some electronic effect like doping, localized states, and so on. So this is at the moment unclear. And the second implication, I believe, is for this PFAS photoreduction, where the colleagues from CA, they showed that actually the milled nanodiamonds, the HPHT nanodiamonds, they showed the best adsorption actually of the PFAS in contrast to DNDs, which did not adsorb them much. And I think it's also could our work can a bit explain that behavior, that in fact in non-conductive DNDs, we think that the adsorption of anionic PFAS is primarily electrostatic, and their zeta potential is kind of stable. But in case of the conductive HPHT nanodiamonds, the zeta potential can be influenced by the adsorbate themselves. So we can, for example, think of some scenario where the adsorbate can induce some charge transfer and also shift of the zeta potential, so it can possibly effectively discharge the particles, and then the hydrophobic interaction may be more efficient. So that's our idea, and it brings me to the conclusion, where I wanted to introduce the hydrogenated nanodiamonds as a not uniform material system with their interfacial and electronic properties depending on the size, internal structure, and the composition. We saw that a kind of minimum size of 2-3 nanometers is needed to obtain this characteristic hydrogenated nanodiamond behavior, like this positive zeta potential and colloidal stability. And the zeta potential is quite probably arising from this charge redistribution and stabilization at the diamond-water interface, which is enabled by the hydrogen termination, but also the structurally disordered surface is important, and this is the result of the thermal hydrogenation. The nitrogen was revealed to plays a key role and be responsible for this tight or specific hydration and adsorption of the CO2. And also we try to disentangle this Raman-like feature in IR spectroscopy, where we showed that it has a different origin, a chemical on DND and the physical on the HPHT nanodiamonds, and the same frequency is just the coincidence. So in the end, I would like to acknowledge my colleagues from the diamond team in our institute, also collaborators from the Institute of Inorganic Chemistry and Biochemistry, Michal Gulka and Petr Ziegler, and also my good collaborator Bohuslav Rezek from the Czech Technical University, and also the external collaborators like Evgeny Ekimov Girard and Jean-Charles Arnaud, and Taro Yoshikawa and Shintaro Ida. So with that, I would like to thank you for your attention.
Stefan, thank you very much for your excellent presentation. It's a lot of new insights. So a lot of very interesting stuff here. Now we are open for the questions, and just to hit up the audience, let me start with my questions. So I'm very intrigued and very kind of happy to hear that these low nitrogen detonation nanodiamonds have been produced. I did not know about that before your presentation, so it was kind of a long, say, anticipated event, but it was not that I knew that it was realized. So can you please tell a little bit more about this very important material, about maybe particle sizes, if there is any difference? It seems they are slightly larger based on Raman than typical detonation nanodiamonds, and also if it's, say, some liquid or ice, say, cooling media are important, because also like ice media also promote larger particle sizes. It's just from that perspective. I don't want to get any proprietary information, but a little bit, it's very intriguing.
Okay. So as far as so regarding the synthesis, I'm sorry, I cannot tell. I also don't know all the details. But for us, okay. So regarding the size, I'm sorry, I didn't share the size. I forgot. We also have a table where we compare the size. So we measured different methods like SAX, which showed actually slightly smaller size of these, and also XRD. If you look a bit carefully, the peak here is a bit broader, so it looks like that according to XRD, the size is also slightly smaller than the typical DNDs, but counterintuitively, their Raman is better. So this is something which tells that this is like a structurally better diamond. I think it's simply that it looks like that the nitrogen somehow contributes to the broadening and the shift of the Raman diamond peak. And when we don't have nitrogen or not that much, the peak can be a bit narrower and not that shifted, even though the particles are slightly smaller. So I think the particles are like 3-4 nanometers in size in general. And here, these DNDs were about, I think, 5-6. So I think definitely these were made with the ice shell cooling. So that's definitely, I agree, one of the way how to control the size. Yes.
Is it a published work on this low nitrogen?
It's submitted. It's submitted. Yes. Okay. So it's not yet published.
Okay. Our next question, who will ask? You can just unmute yourself, please. Or Jonathan Owen has a question, please.
Hi, Stepan. That was a great talk. Some cool materials, and it looks like very careful work, which is great. The one part I don't understand very well is the colloids and the zeta potential versus the XRD size that you quote. So you say the size is like 2 to 3 or 4 nanometers, and then the dispersions that you show are they scatter a lot of light, and they're they're white. So I just wondered, if you measure the size with zeta potential, how does it compare to the size that you're quoting?
Yeah. So I agree that we were completely far from the complete nice dispersion to single individual nanoparticles. So that was unfortunately not the case. Of course, we wanted that, but we were not able to achieve. So with our standard probe sonicator, we got only this milky dispersion, but indeed it was like colloidally stable. So there was, of course, some sedimentation because of the bigger size and the bigger mass, but otherwise the colloids were stable. And I think it's actually quite similar case like the detonation nanodiamond. So that the interparticle bonds are quite strong, and it's not easy to break them or not sufficient by the simple probe like sonication. So the particles, they have, I would say, very, very tight aggregation. So all we got were just some clusters of these individual particles, but I think it still allowed us to measure the zeta potential. And I think here the zeta potential should not depend on the size, basically. I believe that if we get the individual particles, the zeta potential would be the same, basically.
What is the DLS size that you get? I mean, what's typical?
Here it was somewhere about 200 nanometers.
I see. The Z average is the
yes,
DLS
yes.
measured sizes.
Yeah.
I see. I
Yes,
see.
I'm
Okay.
sorry. Yeah.
I have another one, but I'll let somebody else have a turn.
Please go on. Or okay.
Abe, of course, Abe. I was thinking about Abe. Okay, where is he with his questions?
Hi, Olga. So to follow up on
Hi.
John's question and your response, in much the same way, we are then taking a zeta potential measurement of aggregates. They may be dispersed, but it's logical to say that we've got a cluster of our nanoparticles in solution, correct?
Yes.
And therefore, within that aggregate, you're basically only probing that outer layer of the slipping plane of the aggregate.
Yes.
Is that a correct way to interpret that?
I would say so, yes.
Okay. And if there is conductivity, so it's a great connection for the conductivity that you've established for the hydrogenation with the sp2 content. So even with our aggregated, it's almost as if the nanodiamonds are contributing to the overall conductivity, and then during your zeta potential measurements, you've got an agglomerate with a overall conductivity behavior, right? So, and one individual particle you're claiming is conductive, but yet once we have 100 of these guys, the conductivity is still prevalent.
Yes.
Now, is there a limit, and you may not be able to answer this, but is there a limit for the sp2 content along with hydrogenation that turns on our conductivity? Because you showed some data where the 8 nanometer has a lower amount of sp2, and then the 18 nanometer has an increase in sp2, which for myself and maybe for John as well is counterintuitive. I would imagine that the sp2 content would increase with a smaller nanocrystal. And if you could kind of comment on that counterintuitive discovery.
So that's maybe a bit misunderstanding because maybe I messed up a different source of the nanodiamonds. So when we speak about this bottom-up HPHT nanodiamond, and maybe I will just go to my slide. I have okay. So this bottom-up HPHT nanodiamonds are quite special, and if I show you the Raman spectra, you would see that there is no sp2 at all in this size range, right? This 1.3 nanometer is just exception. There we saw some sp2, but it's probably connected with that formation mechanism and so on. But in general, this bottom up, this is very specific type of the nanodiamond. They really don't contain any sp2 surface carbon after the synthesis. What I've shown with this, this one, it's simply a different material and different hydrogenation. So here, this bottom up, still that very specific nanodiamond, it's hydrogenated from the synthesis already. So we didn't do anything with that. But with this top down, with these milled particles, commercial ones, we had to hydrogenate them. So we annealed them in the hydrogen atmosphere. And it's well known that this is the radical process. You actually desorb the original function groups, make the hydrogen radicals, and then these hydrogen radicals hydrogenate the surface. But we always see a little increase of the sp2 carbon after the hydrogenation. So it's this thermal hydrogenation is actually responsible for this increase of the sp2, and that's why we don't see it here. And also, this is the difference also to the bulk hydrogenated diamond, which has the negative zeta potential, but it's typically hydrogenated by plasma. And by plasma, you can achieve very clean, very nicely hydrogenated surface without any sp2 possibly. And that's why it has so different properties. So that's why here we have the two factors for this positive zeta potential, the hydrogenation itself, but also the surface disordered sp2 whatsoever. Did I answer your question?
You did, and I think it was the difference between your top down, our traditional HPHT, and then your bottom up approach with the chloroadamantane. So I got that confused, but thank you very much for the explanation. What's the wavelength of light for your Raman measurements?
Well, this one was UV. It was 325, I think. Yeah. The UV is very useful, and anything lower is with a huge baseline, and if you measure with green, it's already messy.
Stefan, can you please give a little bit more details how you did your hydrogenation? Maybe duration, if you did some, step-like temperature increase and all that, kind of, it's also known, for example, like Tatiana Fegelson published a while ago, her hydrogenation, which was few days with few ramps and so on.
Understand. Yeah. So we don't do anything special. We just we do it in a tube furnace. So we in that study, we always used the same amount of the powder, which was very important to get the consistent results, because of course the degree of the hydrogenation also depends on how much you put in, and there is of course the limiting step is diffusion inside the pores. So that's why we always try to keep the same amount, so then the results were comparable. So that was one thing. The parameters were, I think the heating rate was about 5 degrees per minute. The ramp was typically 6 hours. So we always somehow do the 6 hours, like to be on the safe side, but quite probably 1 or 2 hours would be enough, but we are keeping with the 6 hours. And then the cooling is just we turn off the heating, so it's a natural cooling. And what is also important, we are using the atmospheric pressure of the hydrogen. So it's a pure hydrogen, no dilution of anything.
Not atomic.
Yeah, no. And so it's just pure hydrogen gas at atmospheric pressure, and that's it. Yes.
And what's the amount of nanodiamond you treat in
It
your
was 100 milligrams.
100?
But we can do more, but then possibly we should, here we wanted to see really the exact evolution as a function of the temperature. So here we simply kept everything the same to be consistent and reproducible, but we typically hydrogenate even more, like a gram sometimes. Yeah.
Okay, thank you. More questions, please. Let me
I have another
ask you
one, if that's
another.
okay.
Please.
So I'm still totally amazed that hydrogenated diamond does this charge transfer doping thing. I mean, it's very well demonstrated, but it's bizarre that something that's sort of like a hydrocarbon would do that. So what I'm wondering, I guess, is, the connection between the charge transfer doping and the positive zeta potential makes a lot of chemical sense. But I'm wondering about the more quantitative description. So people have measured the carrier concentration in a power electronics kind of a device, and the total hole concentration in a hydrogenated diamond crystal. Do you have any sense whether how that compares to the extent of charge transfer doping in a nanodiamond? And I'm also sort of curious how that compares to the nitrogen present, because an electron could come from nitrogen and you could store positive charge that way. So have you thought at all? I mean, I don't even know how to relate the zeta potential to an actual charge concentration, but yeah, have you thought about that issue?
Yeah. So that's perfect point. Of course, we thought about that, and so far we don't have any good data about that. Yeah, I totally agree that especially this top-down HPHT nanodiamond it's actually very unique, that it has simultaneously the positive zeta potential, which is unlike the bulk hydrogenated diamond, but at the same time it has this transfer doping like conductivity and the negative electron affinity. So at the moment, we don't have any other characterization, whether like the whole measurement, whether we have the holes as a carriers or even electrons. This we don't know yet. Anything else, unfortunately. But back to that coexistence. So I think also, yeah. I'm sorry. I just wanted to. Yeah, to show this. So yeah, this could be very interesting kind of probe, spectrometric probe of the charge state of the nanodiamonds. So it would be difficult to measure this phenomena on the bulk crystals or on thin layers. But when we have the nanodiamonds, like nanoparticles, it can be easily measured. So at the moment, we are more focused on this phenomenon, like where it appears under which circumstances, because this one, I think it's really relevant to this transfer doping effect. And
you're talking about the Fano resonance intensity.
Exactly. Yeah. Because I think the intensity of this effect should, of course, be should scale with the concentration of the charge carriers created. So that could be kind of a novel kind of probe for how to study these electronic properties of the nanodiamonds. Yeah, and I also wanted to point out this, that here in these two low nitrogen guys, we saw very similar CH stretching pattern, and a very different pattern actually on the DNDs. So it looks like that the nitrogen also somehow modifies the stretching of the CH. So, and I think also the frequencies could be assigned to some particular CH, like CH1, CH2, CH3 on different facets. So it should be also as sensitive to the like prevalence of the facets or like the shape of the particle. And okay, so this is I think the CH on nicely hydrogenated diamond. So this is maybe the reason or the driving force for this transfer doping, charge transfer whatsoever. But still, we see the coexistence of this sp2 features like we see from the Raman. So that's probably that uniqueness of this HPHT nanodiamond, and it explains the conductivity because of this nice hydrogenation and the positive zeta potential at the same time because of this sp2 carbon. That's my view.
I think, you know, so the box, the green box on the right side of the detonation nanodiamond spectrum in green is 2CH, right?
This one.
Well,
Well,
I don't see your cursor at the moment, but anything above 3000 wave numbers, I believe, is from sp2, and you know the 2750 to 3000 is where you find alkane.
Well, that's okay. We had actually this similar discussion with Abbe, and I think here we cannot straightforwardly use this analogy to alkanes or alkanes and to ascribe this stretching to the sp2 or sp3. I would take as a reference some previous, or we take as a reference some previous studies of these hydrogenated diamond facets, where actually I think it's more relevant than the molecules, because when we have the hydrogen termination, I would say the frequencies must be influenced by the substrate. So I think the analogy to the molecules is not very straightforward, maybe. Okay.
Do we have more questions? Maybe one more question. Did you try yet to produce NV centers in low nitrogen DND? Do you think it's suitable material for this purpose? Still, defect structure would be quite high as compared to HPHT, right?
Yeah,
it's still large amount of probably still even stacking faults and still.
That's definitely a good idea, and we are actually working on it a bit. Yeah. Yeah. So it's. I'm sorry, I cannot tell more.
Right. It's kind of natural question, right? And natural thing to try. So, anybody else has some question? If not, then we would like to thank Stepan again for very interesting presentation, and it was great that it was big audience and a lot of questions. Oh, it was somebody with a question. Andrei, no? Sorry. Okay, so It will be great that we all, or many of us, meet in at MRS in the fall in Boston and continue these interesting discussions. Also, I would like to remind our audience, if someone would like to present at Adamas virtual lecture series, please reach us. We'll be happy to arrange you for fall after summer vacations, your talk. So please be,
Okay,
please consider such opportunity.
If I may just say goodbye, I would like to thank you again, Olga. I really appreciate your effort in this sharing, and I really like this platform for the sharing of the result with the community. So really, thanks for your effort and what you are doing.
Yeah, it's very exciting. It's very interesting to do. Okay, thank you everyone, and see you in a month at next lecture. Bye.
Very good. Fantastic talk. Thank you for sharing all that information.
Thank you. Bye.
Great work. Bye.